Hello, my dear students. Last lecture we have discussed about the semi-conservative mode of DNA replication, and it is experimental proof which was provided by Michelson and Sither by using ammonium chloride and heavy isotope of nitrogen. They have just proved that semi-conservative mode of DNA replication. This was proposed by Watson and Crick and proved by Michelson and Sither. This time we will see actually the machinery of the enzyme which is used for the DNA replication. Machinery of enzyme for DNA replication. This topic depends upon the two topics. It's having two subtopics. First, we will discuss actually the types of the enzyme, all the enzymes which is needed for this DNA replication, and after that we will have the process of DNA replication. So, in process of DNA replication, we see there are many enzymes which are used in this DNA replication. The first main enzyme is actually the DNA polymerase. DNA polymerase. This DNA polymerase is actually the DNA dependent DNA polymerase. It is DNA dependent DNA polymerase. It is most important in E-micro. DNA polymerase is actually DNA dependent DNA polymerase. As we know that here the DNA molecule it replicates to form another DNA molecule, and this replication is done by this DNA dependent DNA polymerase. So this DNA dependent DNA polymerase means what? It is actually DNA dependent and also preparing or synthesizing the DNA. This is called as actually replication. Replication is actually self-synthesis. And self-synthesis is the main enzyme which is known as DNA dependent DNA polymerase. And we know that DNA is in helical structure. It is like coil-like structure. Have you seen the rope? In rope there are the two strands. These are just intertwined to each other, and that intertwined each other with hydrogen bonds. They are just fixed to each other by hydrogen bonds. Let me show you here. If this is the DNA, this is the DNA molecule. These are unintertwined, and these are their hydrogen bonds like this. These are hydrogen bonds. This DNA needs to be replicated. So first of all, these hydrogen bonds should be broken down. These are what? These are hydrogen bonds. So for this replication of the DNA, these hydrogen bonds should be broken down. Should be broken down. By this broken down, if we want to break down these hydrogen bonds, we need an enzyme that is called as unwindase. Unwindase, or we can also call it as helicase. So our first enzyme for DNA replication, first of all the DNA polymerase, it polymerizes the DNA, but for polymerization, first thing is to uncoil this DNA strand. These two strands which are anti-parallel, and we have to uncoil this. So because we have to uncoil this, that means we have to break these hydrogen bonds, and these hydrogen bonds can be broken only by this enzyme. The help of this enzyme, that is called as unwindase. So here the first enzyme which is called as helicase. Helicase is the enzyme, or we can say unwindase. This unwindase is the enzyme which helps us in unwinding, or it breaks the hydrogen bonds and unwind the two strands. So this breaks the hydrogen bonds and unwinds the two strands. Suppose this helix or these two strands, they have been unwinded. Suppose, let me write here. First thing, when the helix is unwinded, suppose this is actually this is the DNA structure. Here it unwinds. Here it has been unwinded. Let me show it here. So that means this head will be like this, and this leg will be like this. So there are no hydrogen bonds. There are hydrogen bonds. There are hydrogen bonds. But into this there is no hydrogen bonds. There will be a tension of recoiling because recoiling has been just made unwind by this helices. Now these two strands, they will be in tension or recoiling. In order to release that tension, we are having an enzyme. That enzyme is actually known as topoisomerase. Topoisomerase. It releases the tension of recoiling. Topoisomerase is that enzyme which releases the tension or recoiling or release the tension. So topoisomerase, it prevents the recoiling of two strands. It just prevents the recoiling of the two strands, or you can say it releases the tension of recoiling, or it releases the tension of recoil. So when you are just pulling the rope, two strands of the rope, when you are just pulling it, and then you release that two strands of the rope, that two strands of the rope will again recoil like that same rope. So same is the case here. When you are just pulling this strand from here, this strand from here, and these hydrogen bonds are broken down by helicase enzyme, so there will be a tension for recoiling it. So that tension is released by that topoisomerase enzyme, and these strands will do not get recoiled again. After that, there is a protein. That protein is called a single strand binding protein. It's called a single strand binding protein. So our third enzyme will be single strand binding protein. It can be abbreviated as SSBPs. SSBPs, that is single strand binding protein. When these hydrogen bonds are just broken down, there will be some probability, there will be some possibility that these hydrogen bonds can again be created. So in order to stabilize, there is the instability of these two strands. In order to make them stabilize, these proteins are just used here in order to stabilize these two strands. Is this clear? Now the fourth enzyme is DNA polymerase. The fourth enzyme is the DNA polymerase. The second. Fourth enzyme is DNA polymerase. As we know that DNA polymerase is actually DNA dependent DNA polymerase. In prokaryotes, these are three types. In prokaryotes, these are three types. Three kinds. First kind is DNA polymerase 1, DNA polymerase 2, and DNA polymerase 3. It is DNA polymerase 1, then second one is DNA polymerase 2, and DNA polymerase 3rd. It is DNA polymerase 3rd. This DNA polymerase 1, DNA polymerase 2, DNA polymerase 3. These are having exonuclease, what we call as function. Exonuclease means they can cut the DNA, but they can cut the DNA from the ends. Endonuclease means within the DNA they can cut the DNA, but these are having the exonuclease function. So DNA polymerase 1, it is exonuclease in function. It is exonuclease. Exo means end. Nucleus means to cut. So they are cutting the DNA from the end. You will see in the biotechnology portion what is exonuclease and endonuclease. If you just remember it, that DNA polymerase is exonuclease and it can cut the DNA or it can polymerize the DNA. Sorry, it can cut the DNA from 5 days to 3 days direction or 3 days to 5 days direction. So exonuclease, it can be 5 days to 3 days or it can be 3 days to 5 days endonuclease, exonuclease function. This polymerase first, it is discovered by Kornberg. Most important thing, it is discovered by Kornberg. It is discovered by Kornberg. So that's why this is known as also Kornberg's enzyme. Kornberg's enzyme. By the help of this enzyme, Kornberg has synthesized the DNA molecule. Then next we can have another function of this DNA polymerase 1, that is it can be used to remove primer. It can be used to remove primer. Use it to remove primers. Primers are actually, these are the RNA polynucleotides in a sequence. They form a nucleotide sequence and it is used in the DNA replication. When we see the process of the replication, these primers are get synthesized and then removed from the DNA molecule. So for the first time we will see these primers here. Don't get confused. In the process we will see what is the role of primers. You just remember it that when we want to remove the primers from the DNA replication or new DNA molecule, we can use this enzyme, that is DNA polymerase first. Number next is DNA polymerase second. It is least reactive and help in DNA repair. It is least reactive and helps in DNA repair. Well, after replication, there is some wrongly added bases there and some problems are there in the newly synthesized DNA molecule. That DNA molecules which is wrongly synthesized, it needs to get repair, but the repair is done by this enzyme, that is the DNA polymerase second. It is least reactive and it can help in DNA repair. Is this clear? Also we can see that polymerase 1 and polymerase 3, they are having the exonuclease property. So this is also exonuclease. It's also exonuclease. It can cut the DNA from the ends and in the direction of 3:5. It is 3:5. 3 days to 5 days. It is having the exonuclease function in the direction of 3 days to 5 days. It can cut the DNA in the direction of 3 days to 5 days. And you know that what is the actually polarity? Polarity is 3 days to 5 days or 5 days to 3 days. It can only function in just cutting the DNA in exonuclease, restriction exonuclease or polymerization. It can be used for this cutting of the DNA in the direction of 3 days to 5 days. Polymerase 3 is actually the main enzyme for DNA replication. Most important, it is main enzyme for DNA. This is the main enzyme for DNA replication. Is this clear? This is also known as replicase enzyme. Also known as replicase. and also these are having polymerization activity. Polymerization activity. There is a polymerization activity, and always polymerization is just takes place in the direction of 5 days to 3 days direction. Always polymerization of the DNA or the replication of the DNA, it occurs in the direction of only 5 days to 3 days direction. Now let me knock the board first. We take the screenshot of the board. We'll see other next enzyme. This DNA polymerase in eukaryotes. This was the DNA polymerase in prokaryotes. So we can have these DNA polymerase. These are the DNA dependent DNA polymerase. When DNA is synthesized by its DNA, it is called a replication process. For replication, we need these helicases in order to break the hydrogen bonds or unwanted the two strands to isolate. They prevent the recoiling of the strand or increase the tension of the recoiling. Single strand binding protein, they are just stabilizing these two strands after recoiling. After unwinding, DNA polymerase in prokaryotes is of three kinds: DNA polymerase 1, DNA polymerase 2, and DNA polymerase 3. Now coming to the eukaryotes, as far as NCERT is concerned, there are replication process for eukaryotes, prokaryotes only. There is no mention of this functioning of the enzymes. So let me start here. The DNA polymerase in eukaryotes. If we see, no need to practice it, but for additional knowledge you can have it. But in NCERT it is not written. So it is out of the NCERT. We are just going. But sometimes in the test section, or we can say test papers, or we can say weekend tests or weekly tests, you will be just asked this type of question. So there is a DNA polymerase in eukaryotes. These are of 5 types. Number 1, it will be called as alpha unit. It is alpha, or it can be denoted by like this. This is alpha. Second one is the beta subunit, or we can say beta. Third one is gamma. Fourth one is the sigma. And the fifth one is the epsilon. Like that. These are the 5 DNA polymerases which are found in the eukaryote. It is not in the NCERT. No need to remember it. But for additional information, for additional knowledge, we can remember. Okay. Now there is one more enzyme which is called as primase. Primase is one more enzyme. This primase is what? These are actually DNA dependent RNA polymerase. So they are just synthesizing the RNA from DNA. So that's why these are actually the DNA dependent. So these are just synthesizing the RNA from DNA. So that's why these are DNA dependent. RNA polymerase. These are actually DNA dependent RNA polymerase. What are these? These help us in primer synthesis. This primase, it helps us in primer synthesis. Actually, DNA process cannot be started without these primers. These are RNA primer actually. These are called as RNA primer. But this RNA primer is synthesized by small segment of the DNA, or you can say small sequence of 6 to 7 nucleotide long RNA, long RNA. So it is used as anchor for the addition of DNA nucleotides. It is used as anchor for addition of DNA nucleotides. So this is called as the primase enzyme. Next enzyme that is called as actually phosphorylase. Phosphorylase is what? It just helps in the addition of phosphates. So next is the phosphorylase. Phosphorylase. What is this phosphorylase? This helps in the addition of the phosphate group. Helps in the addition of phosphates. Number next enzyme that is called as nuclease. Nuclease is an enzyme which is used for proofreading. Used for proofreading. When the core molecule of the DNA gets replicated, the nucleus inside the DNA, the proofreading is very most necessary or important process. That proofreading is done by this enzyme that is called as the nuclease enzyme. Is this clear? After that, the last one is the DNA ligase. DNA ligase. When DNA segments are formed during the replication, those segments are need to get joined with each other. That joining of that DNA fragments will be enhanced or will be promoted by this DNA ligase. So join with the fragments of the DNA. It joins the fragments of DNA. So this is all about the what we call as the machinery of the enzyme, or we can say the enzymes which are required for the DNA replication. Is this clear? So I think that is what the concept of this, but in the NCERT you cannot get the separate topic for it. It is included in the machinery of the DNA replication. Many of the enzymes we have learned here, you will not get into the NCERT, but these enzymes we need to get studied in this DNA replication. You just remember it and just prepare the notes for this. We will be in the next lecture of DNA replication, the process of DNA replication. Okay, bye. Take care. Thank you.